what are known as accessory structures of the eye that don't really affect vision, but are involved in lubrication primarily or in keeping dust and dirt particles out of the eye. So this is an anterior view and this is a sagittal view. So on the surface of the eye we have a membrane known as the conjunctiva. You've probably heard of pink eye, which is an infection of that, conjunctivitis. And that membrane, this would be the sagittal view of the eye, this would be the cornea, this would be the upper eyelid. And I thought I had a bit of blue pen. And lower eyelid, so the eyelashes help to keep dirt particles. We know about the blink reflex. And what else happens when you blink your eyelid? You're distributing the tears. All right, so covering the inner surface of the eyelid and covering the surface of the eyeball, both the cornea, the clear part, and the white part, is this membrane known as the conjunctiva. So what is our Latin word for eyelid? The muscle that lifts the eyelid is the levator palpebra. Okay, so palpebral is an adjective for eye. So this is the palpebral conjunctiva on either the inner or the upper or the lower eyelid. And where it's actually on the eyeball itself, it's known as the bulbar. I always think of the babar, the elephant, or whatever. I try to say that conjunctiva. Okay. Then when we look at an on-front view, we can see some additional accessory structures. We have the lacrimal gland. Innervated by cranial nerve 7. And tear ducts passing from the lateral wall of the orbit into the eyeball structure and onto the surface. And so those tears are going to pass from upper outer to lower inner, and this is where we have the drainage. So if we have the drainage system right here, the medial aspect of the eyeball would be dry. And if we had it up here, the lower inferior aspect of the eyeball would be dry. So this is the best location to getting the entire eyeball clear. The corners of the eyelids are known as commissures or canthus. So if we put the nose here, this would be the lateral canthus. You can always tell with the lacrimal gland that's lateral, even if you don't see the nose. So where the two eyelids come together here is the medial canthus. And then this kind of moist area here, if you look, and where you get the sleep in the morning that you dig out of your eyes, is the caruncle. Sounds like a boil. And the two openings here are the punctum that are located right here. You can't see them very well on the picture, but punctum. And they drain the tears into the lacrimal sac. And then they flow down the nasolacrimal duct. Called nasal because it empties into the nasal cavity. Which is why your nose runs if you cry. Overflows the pace at which we normally swallow the tears. Okay, the next set of accessory structures are the, here's just showing the same structure I drew on the board, right, are the muscles that move the eye. And we already looked at these, referred to them when we did the cranial nerves. Okay, so let's do them again. Did everybody see this diagram here? No. Not yet? Okay. I've at least learned to ask.
So the medial canthus is that corner?
Yes, just where, like, the corner of your mouth.
Oh, okay.
If you made this the upper eyelid, this lower, it's just the where the corner of the upper and lower eyelids meet. And if you pull that back, so it was the moist area, that's the caruncle.
Can you see the content?
If you look up close. Okay?
What is the thing in the middle of the?
Is that the
This is just showing the distribution. The blue is just showing the flow direction of the tears. All right, I'll leave that up there and draw another one over here. So again, give you orientation, the nose. And we have the 4 recti muscles. Remember what rectus means? Straight. All right, so coming out at you from the back of the eyeball, we would have the superior rectus muscle, medial rectus because the nose is there, inferior rectus, and lateral rectus. Okay? So you can see from this view, the superior rectus, the medial rectus, the lateral rectus, and the inferior rectus. All right? Missing from this, because the eyelid is missing, is the levator palpebrae superioris, which you could see on the previous picture. So superior rectus is going to help you turn your eye to look up. So both eyelids looking up are going to be superior rectus. Both eyeballs looking down would be inferior rectus. Going cross-eyed would be the medial rectus. However, the 2 lateral rectus muscles do not work together. We don't diverge. So when my lateral rectus on the right side turns to the right, my left medial rectus will follow. Okay? That's what we talk about the eyes tracking together. So what is the cranial nerve that innervates superior rectus? Ocular motor. Medial rectus. Ocular motor as well. And the same for inferior rectus. However, lateral rectus, no. Abductor eye. Abducens. So the lateral, the inferior oblique is going to start from the medial orbit wall and swings underneath and attaches. So kind of this origin would be here, and it attaches to the side of the eyeball. Whereas you can see on that diagram, superior oblique comes in, kind of passes through the trochlea and goes around and attaches to the back at an oblique angle. So superior oblique is going to turn the eyeball. It's going to grab the eyeball from here and rotate it down and out. And inferior oblique is up and out. So looking up here, just using one muscle would be inferior oblique. Looking down here, just using one muscle would be superior oblique. So inferior oblique, I'll write it on the board. Inferior oblique is up and out. Also innervated by cranial nerve 3.
How does the muscle on the bottom move your eye up?
Because it comes underneath and pulls this way. So let me borrow the. So it's going to come like this. It's shown here. It's going to come like here and pull like that. Whereas this one comes in here and pulls like that. Okay. So superior oblique, because of the trochlea here, is innervated by trochlear nerve, so up and out, inferior oblique, down and out. Other way, I'm sorry. I'm wrong. Inferior oblique is up and out. Superior oblique is down and out. Remember this, the cheating one? Looking down and out is superior oblique. Okay, and then what I want to cover today are the basic structure of the eyeball. So on Thursday we can light waves, the energy of the light through the eye, and what structures cause it to bend to allow focusing. So there are 3 layers, or tunics as they are called, that form the structure of the eyeball. The outer layer, you can see here, a middle layer that is vascular or formed of muscle, and the innermost layer, which is primarily where we find our nerve cells, either the receptors or the cells that are going to carry that information out to the brain. So, can I erase the board now? Can you hear me now? So there is one diagram that I repeat several times in your lecture notes. And this time it's a transverse section looking down on the eyeball. So the optic nerve is located medially. So this structure right here is supposed to be the optic nerve, and unfortunately they put it right in the center of the eye. What's the problem with the location of that optic? Well, kind of. You can see it's turned more medially. The optic nerve is in the center of the eye. That's where we focus light. And that's where the macular, the fovea centralis should be. All right? So we look down on top of the eye. We will bring the optic nerve out in this direction, so this would be medial. Well, I can put the cornea more. So the outermost covering has 2 components. It's part of the fibrous tunic, and this portion is known as the sclera. It's what you know and hear about as the white of the eye. It's formed of dense, irregular connective tissue, like many of the capsules of our organs. It obviously has blood vessels. All you have to do is not get very much sleep and look in the mirror. You have all these blood vessels running through your sclera, so it's vascular. And it helps to provide a toughness to maintain the shape of the eyeball. Because if that gets distorted, it changes the focal length, and then vision becomes blurred. It transitions in a region known here as the limbus to the cornea. And the cornea will be the first structure we'll talk about on Thursday that light passes through. Since light is passing through it, it has to be translucent, and it is formed of dense, regular connective tissue. Same as your periosteum, or tendons, or ligaments. Those are not translucent. All right, you can't hold a tendon up and see through it. And yet light passes through our cornea. And the reason that is because the collagen fibers are oriented in a crystalline structure. So they're organized as a crystalline or lattice-like structure. In fact, if you look at a scanning electron view, it looks like dry lasagna noodles, even with the little ruffled edges, and they're laid at right angles to each other. And that provides passageway for light. The cornea should be avascular. Blood vessels can grow into the cornea, which obviously if they grow across the cornea is going to disturb your vision because it's going to interfere with light. There are no blood vessels directly to the cornea, so they have to get their oxygen from the air on the surface. Obviously, if you close your eyes when you sleep at night, that's not available. So then they get their oxygen from the fluid behind them. If you deprive them of oxygen for very long, the fibroblasts making up the cornea will secrete chemicals that attract new blood vessels. Angiogenesis occurs, or the formation of new blood vessels. So I've worn contacts since I was 16, and I went from hard to soft. And I taught a marine biology class in the Bahamas. I studied about fish just so I could get a trip to the Bahamas and teach this and go scuba diving. And so shortly after I landed, I got an eye infection. And there was a college field station. There was no hospital or even a clinic. And so I put these sulfa powder stuff in my eye and patched it up. Your depth perception, you have now when you go scuba diving and a patch over one eye. It was really weird. I could do it on dry land, but scuba diving was just completely strange. So I got back to the States and disinfected my contacts and put them back in, but got the same infection all over again. It was an anaerobic bacteria that dealt well living under the contact. Drove across the country without contacts. I could still read the exit signs. Got to my fiance's apartment, and even inside his apartment, I had to wear a washcloth and then sunglasses because my eyes were so sensitive. So I made an appointment with an ophthalmologist, and I thought he was going to tell me to quit wearing contacts since that was probably the cause of my infection. And he said, no, I want you to wear, this was in the late 80s, 89, I want you to wear the extended wear contacts so that when you take it out, you're not washing it, you're not putting it back in, you're just throwing it away. I was thrilled. I loved extended wear. I decided never to get LASIK when my mom offered to pay for it because I would wear these things instead of 2 weeks, I would wear them for 3, 4, or 5 months. Well, guess what the ophthalmologist saw when I went back to get my eyes checked? Little blood vessels, about 1.5 millimeters into my cornea. Because with the protein not affecting my vision and they felt comfortable, there was not enough oxygen getting to the cornea under the contact lenses, and blood vessels were growing into the cornea. And so he said, you need to take these out every night now, so now I'm going to give you a prescription for ones that you throw away after a day. Forget that after 2 weeks. So they're very fragile. They tear easily anyway. So with the good oxygen supply, the blood vessels just collapse, and they obviously have no blood in them, so they don't affect my vision anyway. All right, so that's the fibrous tunic. We have a case study, keratoconus, I think some of you have signed up for. Keratoconus is a form of astigmatism in which the shape of the cornea is irregular. I mean, that's what astigmatism is. All right, so any irregularity is going to affect the evenness with which the light is bent. And so normally you would have the cornea look like this. So any irregularity is astigmatism. And keratoconus is due to a thinning. The cause itself is not known really well, of one area, and so it can result in a pimple, like from the pressure in the back. But it's referring to the shape of the cornea. All right, the next layer is a deeper layer, closer to the inside of the eyeball. That is not a good looking eyeball. And this is known as the vascular layer. So here's the sclera and the cornea. And the vascular layer is the layer that's going to contribute to the ciliary muscle and the iris muscle that we've talked about already. So these are the three major structures. So there is a vascular layer called the choroid. There are additional blood vessels in the retina, all right, but this is just a specialized vascular layer. And then about 2/3, 3/4 of the way up the eyeball, this expands into smooth muscle, which is the ciliary muscle. And we talked about that being innervated by the ocular motor nerve. And we'll look at its function on Thursday again. And from that, we also see the iris muscle. So we're going to have the ciliary muscle. The iris. And remember, the iris has muscle that's formed in two directions. So we have the dilator, innervated by sympathetic, and it has radial fibers. So when these contract, the pupil gets larger. And then we have the constrictor muscle innervated by ocular motor. And this is the sphincter. So when it contracts, the pupil gets smaller. So sympathetic, parasympathetic. And then we have column-like processes that you can see best on this image here when we do the sheep eye dissection on Friday. And these are structures that make the fluid that we find just behind the cornea. So these, again, are going to go all the way around the eyeball. And they're long column-like structures that extend from the ciliary muscle. And the edge of them is known as, this pattern is known as the ora serrata. But each structure is a ciliary process. And its function is to secrete this watery fluid, known as watery fluid, or aqueous humor. And depending on how much pressure you put on the eyeball Friday, it will squirt out at you or just leak out. Extending from the ciliary muscle to attach to the lens, we have suspensory ligaments. And when we talked about the ocular motor nerve, we talked about how contraction of the ciliary muscle loosened the ligaments and relaxation of the ciliary muscle tightened the ligaments. So we'll go over that again on Thursday. Today we're just doing anatomical structures. All right, so that's the middle layer, or the vascular tunic. So it would be this center yellowish layer. Here would be the ciliary muscle. And if we look at this diagram, go back. So here would be ciliary muscle, the iris, suspensory ligaments, and ciliary process. Yes?
Where's the choroid?
The choroid is this down in here. All right, so if we look at this model, which is a small one, here would be the sclera. The cornea is gone. So the choroid is this brownish layer that contains the vasculature. And then if we lift this up, again, it's small. So then here would be the ciliary muscle, and here would these kind of reddish column-like structures are the ciliary processes. Okay. So we'll have lab to look at this on Thursday, and then we'll do the actual dissection on Friday. So it makes a little bit more sense. All right, so now our last layer is the innermost layer. If we start from the outside, it would be the deepest, or if we start from the inside, it would be the most superficial. And remember when I was describing, when we were talking about the diencephalon, and I talked about the origin of the optic nerve? So here would be the hypothalamus. And I said the optic nerve grew out towards the eyeball and created this cup-shaped structure. And we talked about detached retinas. That would be the lens. So this would be the nervous tunic. This would be the pigmented layer. And this would be the actual photoreceptors and neurons. Okay. So as we draw that here, this portion right here is the pigmented layer. And in humans, it functions, well, most animals, it functions to absorb light that's bouncing around. Because all that's going to do is make multiple images. If you've ever had a camera that did multiple exposures, it makes them blurry. And so we want to absorb that excess light. We don't want it to come in and hit here and then bounce over and make an image over here. Now, animals that move around at night a lot actually have a fluorescent component to that pigmented epithelium where it actually reflects light. You probably noticed at night where you see a cat's eyes, you see this glowing reflection back at you. That's because of that fluorescent area. We'll see it in the sheep eyes. But in humans, this functions to absorb that extra light as well as to recycle the visual pigments that are involved in processing the photons. So when this curves back on itself and makes the innermost layer, these are where the neurons are. And they actually, those are not directly attached to the pigmented layer. So when it's a detached retina, that can be separated rather easily. So these cells out here would be the pigmented cells. You can see that the rods and cones, the photoreceptors, are intimately associated with them. And then, so the outermost layer are going to be the rods and the cones. So I'm just going to hint at that here in the diagram. And then I'll kind of enlarge it a little bit. So this is the neural layer. The 3 layers of neurons. Not attached to the pigmented layer. And the best area to see is an indented area of only cones known as the central fovea. As we move laterally and up towards the ora serrata, we see fewer cones and more rods. So finally, by the time we're way up here, all you have are rods. So let me take this section here, all right? We would have the pigmented layer of cells. So pigmented epithelium. Then we would have rods and cones.
You said the rods and cones are the outermost layer?
They're the ones, yeah, they're closest to the outside.
But not pigmented.
They're right next to so you would have the sclera, the vascular layer of the choroid, pigmented cells. And then, so that's why this is on this side. All right? So rods and cones, rods or cones. And those are photoreceptors. They can only create graded potentials. Then they would synapse with the bipolar neuron. And that would synapse with a ganglion cell that then goes out the optic nerve. So at the central fovea, we have only cones. And then as we move farther in the periphery, we have only rods. So have you ever noticed at night, in my room I have a smoke detector, If I wake up at 2 o'clock in the morning and I look at the smoke detector, I don't see the little light anymore. If I look away, I see the little light saying the batteries are good. If I look at it, I can't see it. Same thing happens at night if you look at stars. You look right at a faint star and it disappears. You look off to one side and it comes, you can see it again. It's really, you try to focus on it and it's gone. And that's because our best line of focus is straight back here on the central fovea. But we only have cones. Colored light has a lot more energy than gray light. So when night light, low energy, lands on that, it's not enough to activate the cones. When it lands over here, it will activate rods, and you will create an image. All right, so that's why at nighttime, if you're trying to see something dim, you need to look to the side and let it activate rods over here. But your best focus is directly straight back, which is color. All right, so we'll stop there. On Thursday, we'll look at the process of image formation. And we'll trace a light pathway through the eye and how it creates that image.